Hello and welcome to the webinar today entitled All Recombinant Proteins Are Not the Same, From Antigens to Avi-tags. My name is David Finkel, Marketing Manager for IMMUNIZATION here at BioTecne, and I'll be moderator for your event today. I'd like to thank everyone for attending today, and I'm happy to welcome our featured speakers for today, Martin Ramsden and John Humphrey. Today's webinar is being presented by BioTechne and supported by Scientist.com. Scientist.com is the world's largest enterprise marketplace for outsourced R&D services. It saves time and money and provides access to innovation while maintaining compliance with an organization's procurement policies. The marketplace features 3,400 plus registered suppliers offering services across multiple industries. Scientist.com operates private marketplaces for most of the world's major pharmaceutical companies and the US NIH. Webinar is presented by BioTechne. BioTechne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Cocris Bioscience, Protein Simple, Advanced Cell Diagnostics, and Exosome DX to provide the scientific research community with a comprehensive and world-class product portfolio of reagents, media, extracellular matrices, small molecules, assays, and instruments. As a market leader of recombinant proteins since 1985, BioTechne and its R&D Systems brand released the first commercially available TGF-beta 1 cytokine. Our proteins are unique and can be used in a variety of applications. Martin and John will discuss our abilities to deliver custom-built protein reagents at bulk levels based on customer-provided project needs, ranging from research use only to GMP grade. Before moving into the talks by our featured speakers, I have just a few housekeeping items to address. Throughout the webinar, we invite you to submit any questions you have for our presenters using the Q&A box at the bottom of the screen. These questions will be asked to the presenters directly following the presentation. And now I'd like to introduce our first speaker for today. Martin Ramsden is the Commercial Development Senior Manager at BioTechne in the Custom Services business. Martin leads the Custom Development Group at BioTechne to forge partnerships with researchers across the globe. Martin has more than 20 years experience in life science research. He received his PhD in neuroscience from the University of Leeds in 2001 and joined BioTechne in 2008. Now, I am pleased to turn the presentation over to Martin.
Thank you very much, Dave. Thanks everybody for being here today, sharing the next hour with us. I'm really genuinely excited for this webinar. I think it's going to be hopefully really useful to you all and also scientifically interesting too. The way it's going to work this morning is I'm going to talk fairly briefly, 10-15 minutes from me, a little bit more background about the companies, the services that we offer, and the way that we try to differentiate ourselves from other CROs. And then I'm going to hand it over to John, who's going to give you a little bit more background about, from a technical perspective, how things work and what the new and hot topics are for recombinant proteins at R&D Systems. So very briefly, just to start off with, I find it important to clarify the way that some of our brands interact with each other. Hopefully, if you're here in this webinar, which is based on recombinant proteins, hopefully you're very familiar with R&D Systems as a company and as a leader in the recombinant protein field. There is still a little bit of confusion with some people regarding how R&D Systems is part of BioTechne and interacts with these other brands. So I just thought I'd try and clear that up right from the start. As Dave mentioned, R&D Systems was founded in Minneapolis in the late 70s. It was a very successful company, especially for recombinant proteins and the Quantikine ELISA product line. So the expertise here is really recombinant proteins, immunoassay development, and we're not always first to mind for antibodies, but also antibody development and manufacture too, which of course those Quantikine kits are dependent. So R&D Systems was a successful company, and about 10 years ago, R&D Systems started to acquire other complementary companies. So for example, Tocris Bioscience in the UK makes small molecule inhibitors. Small molecule inhibitors complement recombinant proteins. Protein Simple, which is an immunoassay automated platform company, perfectly matches with R&D Systems' focus on immunoassay development and ELISA. So as these brands started to come together, there had to be some glue to help communicate this collaboration and synergy, and that's when the BioTecne brand was created. It's an umbrella brand which sits across all of these smaller brands and ties them together. So that's just to clarify how that works. Where it's relevant to services too is that there's two parts. All of these brands offer services based on their individual expertise. And in addition, occasionally we can leverage multiple brands for the same project. So for example, if you're developing a recombinant protein which is going to be used as a standard or a control or an immunogen in an immunoassay, and that's something that you're looking to put on a high throughput immunoassay platform, it's a project that can start with R&D Systems and evolve into immunoassay services at Protein Simple. Here, I really want to try to reiterate how R&D Systems is a little bit different, hopefully in a good way, to some of the CROs and CMOs that you might be used to interacting with. The phrase that I use a lot is that R&D Systems is not a typical CRO. It isn't. R&D Systems is a product development and manufacturing company that also offers services. So the advantage here is, especially for recombinant proteins, is that all of that expertise and experience that we've developed over the last 30 years or so in generating recombinant proteins, all of that is opened up to you as a service project. It's the same scientists that support our retail catalog that deliver the service custom protein projects. The reason that we started to do this, we started to offer services about 7 years ago due to the demand of our customers asking to leverage our expertise, or for asking us to modify a product ever so slightly to meet their needs. So we started doing that, and it's been an extremely fruitful enterprise. It pushes the boundaries of our technical expertise. It makes us better, and it's also led to some really excellent long-term partnerships with our customers. The other thing that I really want to say here is that leveraging an industrial protein development and manufacturing company like R&D Systems, we feel increases the probability of success, because we can use all of that expertise. If you come to us with a request around a protein, most likely we've worked on that protein before in some capacity. We may have worked on other family members for that protein. And we may even have something in the freezer already, which isn't a product. So ultimately, we feel like working with R&D systems not only increases the probability of success for your project, but more importantly, perhaps, it saves time and money in the long term. This is what I'm talking about. This is what I call an industrial approach to developing a new protein. And I admit I might be a little bit biased here, but if you look at a typical CRO, if you go to a CRO and say, this is the protein, I want you to make it, please, and I want you to make it in CHO cells, it's a very linear path. It's often successful or not. And this is where we get into the title of our slide. All proteins aren't equal. If all proteins expressed and secreted and purified, it would be easy, and we'd all be making a lot more progress. That's not how science works, unfortunately. So at R&D Systems, we like to apply a very deliberate, consultative approach. We like to get the scientists together from both parties. We want to understand as much as we can about what you're trying to achieve with the protein in the short term and the long term. What is the intended use? What are the most important specifications for you? And then we can design a strategy accordingly that we would think will have a high probability of success. And part of that is kind of doubling or tripling down. So what I'm trying to show here at the bottom right is if you need a mammalian expression system, we're not just going to stick to one. This is our experience. We're going to probably start with HEC, CHO, and NSO. We may start with 2 or 3 constructs. So immediately, the number of clones that you're going to get when you look at expression is massively greater. Generally, we design this to have an embarrassment of riches at that part, so we can select which clones to go forward. If we're expecting purification issues too, if we think it's going to be a tricky purification, we may want to take 2 or even 3 cell lines into the next phase. So here, ultimately, what I'm trying to show is an increased probability of success, which means time savings, which means cost savings. And again, just to reiterate that, having been involved in the service part of our business for the last 6 or 7 years, saving time is absolutely the #1 priority for the scientists that we partner with. And we think our approach matches that well. It may be sometimes actually that the first phase of what we do may take longer than what you might call a quick and dirty approach. But in the long term, we will save a lot of time. Sometimes the quick and dirty approach is appropriate. It gets you something in your hands to do that first study. The problem is, if it's successful, you then have another transitional period to get to a higher quality product that you need for the next part of the study. So time is absolutely #1. Second to that, I would say, is quality. And by quality, I'm not just referring to the quality of the product, but really the quality and the regulatory support that is provided by the vendor. And I'll get into that a little bit more in a second. And then third is what happens next. If that first study is successful, can you scale up? Can you make it GMP? Can you make different lots of that protein that are highly consistent? And here, quality is important. I'm not going to get into this too much. We welcome quality-based conversations. It's something that we feel we're very strong on. R&D Systems is a high-quality brand in terms of the product, but also in terms of quality and regulatory. If that's critical to your intended use, which it often is, we are All of this work happens in Minneapolis. It's an ISO certified facility. It's FDA regulated. It's governed by stringent SOPs and a rigorous quality management system. And under normal conditions, when people can travel, we absolutely welcome quality audits with open arms. So to focus on proteins a little bit, I'm not going to be modest today. We feel like R&D Systems is the world leader in research use only recombinant proteins. I can back up that claim a number of ways. I think my chart on the left is out of date. I think we've just hit 5,000 protein products. We do this across all sorts of different species and different research areas. The pie chart on the right shows kind of a publication rate. If we take a cross-section of scientific journals and look in the methods section at where those published studies are sourcing their recombinant proteins from, it's always over 40% R&D Systems, which is arguably an underestimate of the market share. So we feel like we have more experience and expertise here than probably any other company. And then in terms of the product quality, our retail recombinant proteins are highly pure. They are typically not released unless we can show relevant biological activity, which implies that the protein is generated and folded in a physiological manner. We have low endo and other contaminants, and we make a highly consistent product from lot to lot. And if you're doing anything long-term, obviously that's very important. And then I should mention too, all of this is Minneapolis-based through R&D Systems. We do also have a Boston Biochem brand in Boston. They specialize in those, in the UPS and everything around ubiquitin, and those harder intracellular targets like transcription factors. So briefly, this is how our service partners present to us. It is very much generally a problem and solution situation. The problems come in, I can break the problems into sort of 3 phases: early, mid, and late. So for early, the customer may be coming to us because they don't have the resources or the capability or the expertise to make a recombinant protein. Their expertise maybe is something else, and they just need that piece. They need to outsource that piece. Or they may be trying to make the protein and they're running into problems. The mid section of the workflow is generally customers have made a protein, but they can't hit one of the specs. Often this is purity or endotoxin based, or they're seeing inconsistencies between lots that they need to fix. And also we have an increasing number of customers that come to us. They've made the protein themselves or through another partner. They're happy with the product in the small scale, but they need a vendor that has a higher scale capacity, or they want conversion to GMP for their intended use, which only a certain number of companies are capable of. So not surprisingly, the services that we offer around proteins mirror our own expertise that supports our product development and our catalog. It also mirrors the proteins. So if you need a protein from scratch, we can do that, as I talked about right at the start. If you need specific help with expression or purification, we can do that too. Importantly, modification. If you have your own protein, or more often, you're using an R&D Systems protein and you like it and you're happy with it, but you wish that you could just change one of the specs. Maybe for your intended use, you need lower endo, or you need a different tag. It's an FC and you want HIS or something like that. We can often modify the product for you. And like I said, if you need GMP proteins for cell and gene therapy or other uses, we can certainly support that in our GMP facility. So I only have a couple more slides, I think, before we get over to John, but this shows what you would expect if you partner with us. Again, we call ourselves consultative. We really want to understand as much as we can what you're trying to do. Again, the reason that we do that is we want you to benefit from all of the expertise. So the more we know about your project or the technical challenges that you face, the more we can address them. We also want to think about time. And the more we know about the project up front, we can do things in a certain way that would save time in the long run. The way it works, what you would get is a formal proposal. We call it a statement of work. It will show all of the things that we're going to do that we've agreed on, the things that we're not going to do sometimes. It will show you the timeline and the deliverables. And once we have that locked down, it'll have an approximate price. When we have that locked down, we will lock the price in and quote it. And then, of course, if you initiate the project with the same project manager that's been helping you so far, that dedicated project manager to your study will keep you regularly updated throughout the progress of the project. I'm not going to spend too much time on this because I want to get over to John. I think I'm going a little bit long. This is a case study. Very quickly, this customer came to us about 1.5 years ago. They had 3 problems. They had poor yield. They had lots of purification issues. The intended use was ex vivo therapeutic. So the purification was already difficult, but they couldn't tolerate anything like nickel or heparin or anything else that might be considered a residual. So we had to proactively plan around that. And then they needed the product to be GMP. So in short, we massively improved the yield. We fixed the purification to their requirements. I think we made 2 or 3 RUO lots, a verification lot, and then the 3 GMP lots. And I'm delighted to be able to say that that product right now is in a phase 1 clinical trial. And also of note, during the end of that study, the end phases of that study, we also partnered with that company to generate a DMF, a drug master file, that supports their regulatory submissions to the FDA. This is my last slide, just a quick advertisement. This is a new service. If any of you guys are purchasing large numbers of R&D Systems proteins, so I'm talking about dozens or even hundreds of proteins, and you're buying them in probably small aliquots, and then you're plating out all of those proteins, most likely for some sort of high throughput screen. Talk to us about it. This is something that we've had demand for. We now offer it as a service. The last one we did, I think we delivered 400 different proteins, plated out, frozen liquid, in duplicate in 96 well plates. So the advantages are you don't have to do all of that busy work. There is some cost savings in the protein purchase. It's set up by our professional product finishing group, so you get potentially less variability and better data. So if that's something that's new, it's exciting. If that's something that sounds appealing, please contact us and let us know. So I think that's it from me. Again, thanks very much for joining us. Any questions, please reach out in the chat or contact us afterwards, and I'll hand it back to Dave.
All right, well, thank you for that very informative presentation, Martin. Quick reminder, if you do have questions, use that questions box down below. And now I'd like to introduce our second speaker. John Humphrey is the Director of Molecular and Cellular Biology, joining R&D Systems in 1988, and developed key processes for enhanced recombinant protein expression and secretion. He has led teams in molecular biology, cell culture, and protein production, overseeing the expression of over 20,000 recombinant proteins and the establishment of thousands of stable lines for antibody screening and reporter assay development. And now I'll turn it over to John.
Thanks, Dave. I'm glad to be here and sharing this with you. In the respective time, I'm going to be racing through this data rather quickly, but just a reminder that this is being recorded, and please send in your questions as we move forward. So this is just a reiteration of what Martin had talked about as far as the content goes, and it's pretty well self-explanatory. I think we've already touched on this very much with the multiple platforms and large content that we have, particularly our non-catalog content that we've developed over the years. For our customers, you know, most of you listening to this webinar have applications in this area, and in bioprocessing. Cancer research, of course, is still a huge area, especially immune oncology. Cell therapy is growing rapidly, and then also supplements into media and with cytokines and growth factors are key. There's one other application, of course, that's a little bit more forward in our thinking the last few months, and I'll address that shortly. So in order to get to a high level of quality, our in-house development and our bioassay development has really been key to this success. And we have a lot of content to leverage in the bioassay area, and this is just a smattering of the different types of assays that can be used to show relevant biophysiological activity. Most of these assays were developed in-house by our scientists. Everything from neuronal outgrowth, chemotaxis, specific reporter assays, and different ways that we can assure quality from lot to lot and reproducibility from lot to lot. We also do a regular competitor testing. We want to make sure that we calibrate our products with the competition. And so we put these, and that's where our assays help us as well. By having sensitive bioassays, we can look at our customer offerings and make sure that we are equal to or better than specific bioactivity in certain areas. And this is an example for our spondin. Consistency, of course, and Martin touched on this as well. We use reference lots, and we calibrate our subsequent lots back against those reference lots. And if something doesn't follow the normal bioactivity, we won't release that lot. And so this is just a way to assure you as a customer that over time, the products that we produce, even if it's 4 or 5 or 6 years down the road, they're going to behave the same way that they did in the past. Here are some of the things that we've received from customers when we're looking at designing proteins for them in their projects. And these are the questions that we like to know. What is your intended use for your protein? Is this something that's going to be used in a binding screen? Is it something that's going to be used for a bioprocessing application? The intended use is very important in how that protein is produced. Martin also touched on the different host cell platforms from E. coli through mammalian cells, utilizing those different platforms. We have that at our disposal. The domain structure of the protein is very important as well. And so sometimes we have customers that are looking at just expressing portions of proteins, and we like to do some analysis on the structure of a protein and make sure that we're not interrupting potential bioactivity by changing the domain structure of that protein. We look at disulfide bonds to make sure that we don't end up with free cysteines that could cause aggregation and things along that line. And then we also look at where a tag, if a tag is needed, that should be located. The diagram to your right is a project that we had a customer asked us for, and this is IL-12 is a heterodimeric cytokine. And the issue with it is when you co-express the subunits, you don't always get a 1-to-1 ratio of those subunits. Sometimes you'll get an overabundance of a homodimer versus a heterodimer. And so it's a little more difficult to scale. We made this linked version of IL-12. And so the linker portion was very important to be able to allow both subunits to form the appropriate structure and folding to be active. And so this has helped us to be able to scale IL-12 immensely over the naturally associated subunits. So when you're thinking about antigens for immunization, there's a lot of different approaches that people can use, and it depends on what you're going to be doing with what your intended use is. So for flow cytometry, for example, for GPCRs where they're a multipass protein and there's not always a lot of extracellular domains available, what we have done in a lot of our antibody development for our customers is use transfected cells to immunize with. This allows the receptor to be presented and in a natural way. And we've found that it's a way to get higher quality flow cytometry antibodies that will recognize the conformation. And sometimes it's interactions between other proteins that cause this effect as well. We have, a lot of different targets and so forth for antigen use. We've focused on mostly full-length proteins, but we've used domains as well. And again, depending upon what the use of your antibody is, we've found that in most cases, unless you're looking for a specific epitope, as you see in the peptide column, it's nice to have active, properly folded antigen to produce quality antibodies. Peptides, on the other hand, are great for phospho-target specific epitopes or differentiating between conserved protein families. And just yesterday, we had a press announcement of our first llama body, and this is against SARS-CoV-2 RBD, and it is a blocking antibody against that. So it's an exciting thing. We just launched it on our website, and that's a new product, and we're expanding that area as well. So what's going on in proteins for us? What are most of our customers asking us about? Avi-tag is probably one of our hotter areas that's in demand, and Avi-tag uses the BIOASSAYS ligase, recognizes a specific sequence, and when the BIOASSAYS enzyme is expressed with your target protein, it will transfer a biotin to that target sequence in a specific manner. That allows you to orient your protein for screening by using a strep-avidin-coated surface. This is an example of some SPR data that was obtained for CD155, and you can see a nice sensitive interaction with TIGIT with the Avi-tag CD155 coated on the surface. We currently have about 80 Avi-tag proteins available, and we're adding them on a monthly basis as well. This is the hot topic, and I guess I suppose none of us ever thought that immunology would become a spectator activity, but it's kind of become that. We hear about it every day in the news, and virology and immunology now is in everybody's vocabulary. And so all of us, I think, all of you attending, have pivoted in some way to address the SARS-CoV-2 issue. We currently have about 21 characterized coronavirus proteins available, but we're rapidly adding to that as quickly as we can. And I'm not going to go through the whole list of coronavirus proteins. I think you're well versed in that based on the audience here. So this is a busy slide, but it shows where we focus to start releasing our products. And what I'd like to point out here is not only have we just released a RBD, or receptor binding domain product from SARS-CoV-2, we've made it in a number of different cell lines with a number of different tags, just for the plethora of applications that our customers have asked for in these types of proteins. This list contains all spike protein related products, and that's growing rapidly too. But as I'll show you in a couple slides coming up, there is a difference from cell line to cell line. This is some data that we use to qualify our spike proteins, and it shows both the RBD with an FC tag and the RBD with a His tag binding to the coronavirus receptor ACE2. And also some SPR data validating this as well. We've tried to characterize these as thoroughly as possible because we do see differences from vendor to vendor in performance of these viral proteins as well. Particularly with all the secondary modifications that are involved on the spike proteins. Here's an example of the S1, so the full S1 domain binding to ACE2 as well, very similar to the RBD only. And again, the SPR data that backs that up as well. So we're following a theme with our spike proteins in being sure that we can assure the customer that they do what they're supposed to do and they bind to the appropriate receptor. We also have the ability to characterize through SEC the form of the protein. And this is something with the spike protein that we do as well, because we have different multimers that form depending on the length of the portion of the spike protein, the length of the protein, and the system that it's expressed in. And this shows a trimer from two different cell lines, CHO and HEC. And you can see that there's a little difference in the molecular weight, and I'll show you some more data on that as well. But both of these run as a trimer. So we have some expertise in glycobiology, and we have a scientist that has put together a really nice toolbox on characterizing glycans. And we wanted to see just what is different about spike proteins that might be expressed in the different cell lines. So you can see in the diagram below, we labeled CHO, SF21, and TN5 cells and hex cells. They were completely, you can see on the diagram too, where the RBD proteins expressed in these cells only contain simple core-1O glycans. The labeling of the HEC293 cell was resistant to ENDOF, which removes core-1O glycans. And neuraminidase resulted in a significant increase in labeling of the protein expressed in HEC cells, but not in the other cell lines. So that the O-glycan, and this suggests that the O-glycan on HEC cells is highly sialylated, and as you can see in the diagram above. Next slide. He also did some analysis on N-glycans, which was very interesting. The results of this study which you can see in the different colors, red for the complex and green for the high mannose. So complex N glycans are found only on CHO and HEC expressed RBD His. As you can see, it's mostly stained red with a little bit of green as well. But in the insect cells, not surprisingly, you just see high mannose and glycans, and then a very, very small percentage in the CHO and HEC, but in the insect, that's the prevalent. So this is kind of a summary of what these two experiments showed. Is in the RBD His expressed in CHO has complex N glycans and a core 1 O glycan. In HEC293 cells, it's different because both the N and the O glycans are sialylated. And in insect cells, SF1 or TN5, it's predominantly high mannose with the N glycan and the core 1 O glycan. So the protein, as you think about it, is the same, but it's not really the same based on the post-translational modifications. This is a growing list of our ACE2 proteins as well that fit into this coronavirus area of research. We're in the process, we should have available shortly some Avi-tag labeled ACE2 proteins, human ACE2 for binding studies. We also have positive and negative controls of different species as well. The other proteins that are quite important in coronavirus biology, particularly since I think there needs to be a focus on antiviral development as well. And so what are some of the proteins that would allow you to interrupt the viral cycle and stop the virus from multiplying? So I think the development of additional antivirals, which is obviously needed as well as a course of action in battling this infection. These are two examples, because the papain-like protease has functions that recognizes sequences and cleaves them in the viral sequence to activate and release other proteins. And the C3L main protease, there's about 12 non-structural proteins that need to be activated for viral replication. So if you can interrupt and block the action of these proteases, there's a good chance that it will be an antiviral effect. Here's a list of the products that we have available of the two proteases that I just mentioned. And we're also working on some other protease targets as well. And we've had customer inquiries in some of the non-structural proteins that could be key to viral reproduction as well, or replication. This is, as you recall on Martin's last slide, he mentioned proteins in a plate. And the way that this product came about was a few years ago. We had all these proteins at our disposal, and as immuno-oncology was emerging, we thought that why not do a protein screen and see if we can identify some novel protein-protein interactions. And so we did a fairly massive protein in a plate screening and looked for binding partners amongst the different proteins that we had at our disposal, and did uncover a couple things that we did end up publishing. But this inventory in the immune-oncology area is quite rich. So we have a growing list of proteins that are available, and we can produce these and modify them in ways that would apply to your specific application as well in cancer research. What we're shortly will be releasing, and they should be on our website anytime soon, are reagents that our customers asked, well, we're engineering in cars and putting them in either T cells or NK cells. how can we tell the efficiency of the transfection of these cells before we put them back into a patient? And so we've labeled some cytokines with fluorescently tagged proteins that are to CAR targets, such as the CD19, BCMA, and some of the other key CAR targets. We've started with the T cell targets, but this could be applied to NK cells as well. And so this is something that could also be done on a custom basis for your particular CAR. If you're developing something and you want to monitor the efficiency of your CAR expression. I'm getting to the end of the data here. These are a couple proteins that are a little bit still controversial because their effect has been shown in mice as far as bioactivity goes. IL-39 was shown to mediate inflammation in lupus in mice, but it really hasn't been confirmed in humans. But we do have a product, and it was very difficult. We just got nowhere in trying to express these subunits separately and have them associate. So we made a linked version and with an FC tag, and we were able to remove aggregates, increase the yield, and have a protein where we could actually measure the bioactivity. ILY is another protein that in mice, it suppresses diabetes effect. It was published by the Scripps University of Pittsburgh group, I believe last year or the year before. This again is something that has not been clarified in humans, but it shows ways that we can actually engineer proteins to come up with either unique activities or activities that can be assayed in some way. And Martin already touched on this too, but I just want to reiterate the GMP process that we are expanding as well. Our new facility is on track to be coming online towards the end of the year. We currently have, this is an expanded standalone facility that we're expanding our GMP production capabilities in. Our GMP facility is housed in our existing main campus facility. But the level of quality that he already talked about is listed here. These are some of the quality testing specifics that we do on our GMP proteins. We also have instruments in development where we can optimize strains that are used for expression. We can optimize production and scalability of your particular ex vivo GMP target. And so we're getting more and more custom projects that follow along the lines of GMP development as well. In addition to our on-house GMP facility, we have a dedicated animal-free facility where we're able to produce proteins in a controlled environment. And everything's traceable, and we have a number of different products available with that type of designation as well. We can do everything from small scale to master supply agreements, custom vial sizes, and our catalog products have grown to about 40 GMP cytokines and growth factors. That kind of wraps up the data, and I'm going to turn it over back to Dave, and we can address any questions that may have come in. I know that went quite quickly, but I wanted to leave some time for questions.
All right. Well, thank you, John. It was very informative. Before we begin the panel discussion, I would like to remind attendees that they can submit the questions in the Ask Question Q&A box below. So do that. We have a few come in, and I will ask them, and John or Martin, you first come, first serve. Do you offer low endotoxin services?
John, I can do that one, I guess. We do. It's reducing endo is one of the common modifications that we get requested for. Probably conjugation, mainly biotinylation, and then low endo are what people are asking us to modify our retail products. Low endo is something we've done a lot of over the last year or so, actually. So they can get that down over 100 to 1,000-fold lower than the typical catalog spec. So yes, it's something that we very much can do. So if you are putting a protein into an animal for some sort of in vivo study, then we can help you with the endo for that.
Would I be right in thinking you're not would remove the endo, but you'd probably have to go back and re-engineer or do something with the cell line at the beginning to make sure it's just a low endo?
It goes in there's two different paths, I believe. So obviously we'd want to talk to the customer and understand, which protein and what they're doing. But yes, we can either, depending on the spec, take the lot and effectively repurify it to get the contaminants even lower. Or if you really are looking to lower it, I think we can go down to around 1 EU per milligram. Then you start over. Yeah, you make a new lot under even more stringent, clean, new reagents, all that kind of stuff. So there's two paths to it, I believe.
Gotcha. It's nice to hear that you what is the customer spec? It's what you go by. What does the customer want? So it's nice.
Customer spec is king.
Yeah. Okay. So another one is, can you introduce disease-related mutations into the proteins?
I can take that one, Martin.
Yeah, that's one for you, John.
Yeah, we do that quite often. Actually, we have had, and this disease-related mutations is one thing that's very important. But it's also, speaking of mutations, there's also services that we have had several times where customers are doing scanning mutagenesis screens on their proteins. So they'll send us, or they'll have us produce, let's say 24 different mutants, just changing targeted amino acids to change the characteristic of the protein for whatever they're doing. So we do those kinds of things either on a small scale, like you mentioned, Dave, for a disease-related mutant, or more of a high throughput, large scale mutagenesis where we're doing a lot of different constructs, just smaller quantities for them to evaluate.
And then you mentioned small scale, but so if something works on a small scale, what about ramping it up to bulk? Some people have bulk needs, and imagine you're ready to do that if they ask.
Right. So we do have the ability to scale things dramatically, and for example, now we're producing some of the SARS-CoV-2 proteins at gram levels, and so we have the ability to go from just 100 microgram quantities for evaluation to many MIGs and up to gram quantities of different targets. It's all protein dependent, of course.
Excellent. Okay, well, keeping in line with Martin talking about time is so important, I think we only have time for one more question. And could you make a specific lot of our protein, the customer's proteins, to be slightly different in the specifications? So taking their protein and doing something with that?
Yeah. So we've had this question come in as well, and I'm trying to remember of a particular spec, but we have had people come to us where they either want a different modification or they want the spec different than what our catalog product is. And depending on what that is, and we have a strong analytical core that can do the analysis and testing to go even further to make sure that we can meet the additional spec that would be introduced into that. So it might be a modification, it might be something else that the customer is interested in that they need.
Yeah, just to add to that, Dave, there's the change of specification that John's talking about. I've seen a handful of projects too where the customer is happy with the product, and they're happy with the lot-to-lot consistency of the product. But because each time they receive a new lot, they have to qualify it and do bridging studies, it actually works out to be more economical and more efficient to have us make a dedicated lot that they can feed off maybe for a year or 2 years, so they don't have to keep qualifying new lots. And that's something that appeals to a lot of people in the long term.
Excellent. All right. Well, you know, to me, it sounds like it starts with that conversation, what somebody needs, what the capabilities are, and then having that conversation and you know, moving forward if it's working for everybody. And this has been a great webinar. I thank you to Martin and John, scientist.com. Great job. And we're at the top of the hour here. And so I'm going to say thank you for attending. And if you have any questions, reach out to us at BioTecne and we can start that conversation. So thank you, everyone.